Welcome to the Deep Dive, where we cut through the noise to bring you the most essential insights. Today, we're plunging into the really intricate world of pharmacology. Ever stop to think what truly happens to a drug after you take it? I mean, it's way more complex than just pill in, effect out. Our mission today is to unpack the fundamental principles, how anesthetics and really lots of other medications journey through your body, how they work, and how they eventually make their exit.
Exactly. And we're here to equip you with let's say, a more robust understanding of the science behind it all. We want to go beyond just the definitions, really get into the how and why of drug behavior. We'll touch on everything from how they find their targets, how they spread out, get broken down, and eventually, leave your system. And yeah, you might actually discover some surprising ways drugs can get sort of trapped once they're inside you.
Okay, so let's kick things off right at the beginning. How do drugs actually interact with our bodies on that, cellular level?
Right. This is where we get into agonists and antagonists. It's actually pretty fundamental. Think of a receptor on a cell as a kind of lock. An agonist is like the key that fits that lock and turns it. It activates the receptor, produces an effect. There are different types, right? Full agonists turn the key all the way. Partial agonists, well, they only turn it part way. And then there are even inverse agonists. They bind to the same lock, but actually produce the opposite effect of the agonist. Kind of turns things down instead of up.
Okay, so agonists are the activators. What about the antagonists then?
So an antagonist is interesting. It fits into the keyhole too, but it doesn't turn the lock. It just sits there. It basically blocks the agonist from getting in and activating the receptor.
It just blocks the spot.
Exactly.
Yeah.
Now, how it blocks matters. A competitive antagonist is like, well, imagine someone keeps trying to put the wrong key in the lock. If you have enough of the right key, the agonist, you can eventually push the wrong key out and open the lock. So you just need a higher dose of the agonist.
Okay, you could overcome it with more drug.
You can, but then you have non-competitive antagonists. These are different. It's like someone welded a plate over the keyhole. Once that antagonist is bound, it doesn't matter how much agonist you add, you just can't activate that receptor anymore. The maximum effect is reduced.
That's a really clear distinction.
Yeah.
So, okay, the drug finds its lock, maybe turns it, maybe blocks it. But once it's in the system and ready to act, how does it spread out? Where does it go first?
Good question. That's all about distribution. So after a drug gets into your bloodstream, say from an IV, it doesn't just stay there. It starts mixing with your body tissues really fast. And the first places it goes are the organs that get the most blood flow. We call this the vessel-rich group. Think heart, brain, kidneys, liver, vital stuff.
Because they get the most blood pumped to them.
Precisely. They have the highest perfusion, so they see the drug first and fastest. We even have ways to measure this, like rate constants, tracking how quickly it moves between blood and tissues.
Okay, vital organs first. Now here's a term that always sounds super technical but seems really important. Volume of distribution, or V-day. What's that telling us?
Yes, VR day. It sounds technical, but the concept is actually quite powerful. V is essentially a theoretical volume. It tells us how widely a drug seems to spread out in the body relative to its concentration in the blood. Basically, where does the drug like to go?
Okay.
And here's the key idea. We compare it to total body water. That's about, 0.6 liters per kilogram, or maybe 42 liters total in an average adult. If a drug's V is larger than that total body water.
So it's like it's filling up more space than just the water.
Exactly. That tells us the drug is lipophilic. It's fat-loving. These drugs don't just stay in the water compartments. They dissolve readily into body fat. They spread out a lot. And because they spread out so much, you generally need a higher dose to get enough concentration in the plasma to have an effect. Propofol is a classic example. Huge V.
Right, and no propofol. Okay, so what if the V is smaller?
If the V is less than total body water, the drug is hydrophilic water-loving. It doesn't really go into fat much. It sticks to the watery parts, like the blood or the fluid outside the cells. This means you usually need a lower dose. Neuromuscular blockers, the drugs that cause muscle relaxation for surgery, they're often hydrophilic, restricted mostly to that extracellular fluid space.
So lipophilic means large V-day, higher dose needed. Hydrophilic means smaller V-day, lower dose.
Got it. You got it. And yeah, most general anesthetics tend to be pretty lipophilic, which explains their large V-day.
That makes a lot of sense. But, okay, what if the drug wants to get somewhere, but something in the blood itself is holding on to it? This brings us to protein binding, right?
Indeed. Protein binding is another big factor. See, many drugs, once they're in your bloodstream, don't just float around freely. A significant portion actually sticks or binds to proteins in your plasma. Albumin is a big one, often binds acidic drugs. Then there's alpha-1 acid glycoprotein, which tends to bind basic drugs.
So they're kind of stuck to these proteins.
Yeah, temporarily. And here's the crucial point. It's only the free, unbound fraction of the drug that's actually pharmacologically active. Only the free drug can leave the bloodstream, cross membranes, and get to the receptors to do its job.
So the bound part is just inactive.
Essentially, yeah. It's like it's in reserve. And interestingly, there's often a link. Drugs with high protein binding also tend to be more lipid-soluble. They like fat, and they like sticking to these proteins.
Man, the body is just this incredibly complex balancing act. Okay, so a drug gets in, finds its target, does its job, but it can't stay forever. How does our amazing body actually get rid of these substances?
Right, time for cleanup. That's metabolism. The liver is the main player here, definitely the powerhouse, but other tissues chip in too: lungs, kidneys, the gut lining, even the placenta. The main goal of metabolism is usually to take a drug that's active and often lipid-soluble, hard to excrete, and convert it into something water-soluble and usually inactive.
Making it easier to pee out, basically.
Pretty much, yeah, or excrete in bile. This usually happens in 2 steps. Or phases. Phase I reactions involve things like oxidation, reduction, or hydrolysis. They basically chemically modify the drug molecule. The big enzyme system here is cytochrome P450, especially an enzyme called CYP3A4. It's super abundant in the liver and chews up a lot of drugs, including many anesthetics.
Is that the grapefruit thing?
That's exactly the grapefruit thing.
Right.
Grapefruit juice can inhibit CYP3A4, messing with how certain drugs are metabolized. Now, hydrolysis is a bit different. That often happens outside the liver, like in the plasma, using different enzymes. Think remifentanil, acetylcholine, esmolol. These get broken down super fast in the blood. Their action is really short.
Okay, so that's phase 1. What's phase 2?
Phase 2 is conjugation. The body basically sticks a larger water-loving molecule like glucuronic acid onto the drug or the phase I metabolite. This makes it much more water-soluble, really flags it for excretion. One important note: infants, especially newborns, don't have a fully developed conjugation system yet, so some drugs can be more toxic to them.
So the liver's this processing plant. How efficient is it? Does it clear some drugs better than others?
Absolutely. We talk about extraction ratios to describe that efficiency. If a drug has a high extraction ratio, it means the liver is really good at grabbing it from the blood and metabolizing it on the first pass-through, like a large percentage is removed each time the blood flows through. For these high extraction drugs, the limiting factor isn't really the enzymes they work fast enough. It's more about how much blood is flowing to the liver. Less blood flow means less drug delivered, so less clearance. It's perfusion dependent.
Okay, so blood flow is key for those. What about low extraction drugs?
For drugs with a low extraction ratio, the liver isn't grabbing much on each pass. So for these, clearance is much more dependent on the actual activity of the liver enzymes and also how much drug is free versus bound to protein. Changes in enzyme activity, like induction, where the liver makes more enzymes, can significantly speed up their clearance. These are capacity dependent.
Right, so it's this interplay. Blood flow for high extraction, enzyme capacity for low extraction. And it's not just the liver handling elimination, is it? Kidneys are involved too.
Definitely. That's renal clearance. The kidneys are like these amazing, sophisticated filters. They use a few processes. First, there's glomerular filtration, where water and small molecules, including drugs, get filtered out of the blood. Then there's active tubular secretion, where the kidney cells actively pump certain drugs from the blood into the urine. And finally, passive tubular reabsorption. This is where some drug can actually sneak back from the urine into the blood.
Wait, it gets filtered out and then comes back in?
It can, yeah, especially if the drug is highly lipid-soluble. Remember, lipid-soluble things cross membranes easily, so it gets filtered, but then it can just diffuse back across the kidney tubule cells into the bloodstream. This means very little of the unchanged lipid-soluble drug might actually end up in the urine.
So water-soluble drugs get cleared by the kidney more easily?
Generally, yes. And urine pH plays a role too. For example, if you make the urine more alkaline, weak acid drugs become more ionized and trapped in the urine, so they get excreted faster.
Fascinating. Yeah. Okay, knowing how long a drug sticks around is obviously crucial, especially if you're giving it continuously, like in an infusion. Let's talk about elimination half-time.
Right. The elimination half-life, often written as T12, it's a pretty core concept. Simply put, it's the time it takes for the drug concentration in your plasma to decrease by half after you've given a single quick dose, like an IV bolus. It's related to both how widely the drug spreads out, that volume of distribution we talked about, and how quickly your body clears it. It's useful because it gives us a rough timeline. Generally, we figure about 4 to 5 half-lives need to pass for almost all the drug, like 95, 97%, to be eliminated from the body.
So 1 half-life, 50% gone. 2 half-lives, 75% gone.
Exactly. 3 gets you to 87.5%, 4 to about 94%, and 5 is nearly 97% gone. It gives you a predictable decay curve.
Okay, that makes sense for a single shot.
Yeah.
But then there's this other term, context-sensitive half-time. What's different about that? Why do we need another type of half-time?
This is a really important distinction, especially for anesthesia with infusions. The regular half-life, T12, describes elimination after a single dose. Context-sensitive half-time describes how long it takes for the plasma concentration to drop by 50% after you stop a continuous infusion.
Okay, so it's specifically about stopping an infusion.
Yes, and here's the crucial part. The context-sensitive half-time changes depending on how long the infusion was running. It generally increases the longer the infusion goes on.
Why? Why does the duration matter?
Because during a long infusion, the drug doesn't just stay in the blood. It distributes out into all those other compartments, muscle, fat, etc. The longer the infusion, the more these compartments fill up. When you stop the infusion, the drug starts leaving the blood, yes, but it also starts slowly leaching back into the blood from those saturated tissues. This coming back effect slows down the drop in plasma concentration. So removal from plasma isn't the same as removal from the whole body after an infusion.
Wow, okay. That really highlights how complex drug movement is. Now, this is where things get maybe even more interesting. Why do some drugs seem to almost get stuck or accumulate in certain body areas? This leads us to ionization and PK.
All right. Yes, this is a fascinating bit of chemistry that has huge physiological effects. So many drugs are weak acids or weak bases. This means they can exist in 2 forms. An ionized form, which has an electrical charge, and a non-ionized form, which is uncharged.
Charged or uncharged? Yep. Okay.
The ionized form is generally more water-soluble. Because it's charged, it has a hard time crossing lipid cell membranes. It's also usually the inactive form and is more easily excreted by the kidneys. The non-ionized form, however, is lipid-soluble. It's usually the pharmacologically active form. And because it's uncharged, it can easily slip across cell membranes, think the blood-brain barrier, the placenta, getting into cells to be metabolized by the liver.
So non-ionized gets you across membranes and does the work. Ionized gets stuck and washed out. What decides which form it's in?
2 things. The drug's own PK, which is basically a measure of its inherent acidity or basicity. It's a fixed property for each drug, and the pH of the fluid it's dissolved in, like your blood or urine or stomach acid. The PK is the specific pH value at which exactly 50% of the drug is ionized and 50% is non-ionized.
The halfway point.
Exactly. And the relationship is predictable. If you put a weak based drug into an environment that's more acidic than its pKa, more of it becomes ionized. Put a weak acid drug into an environment more basic than its pKa, again, more becomes ionized. Take lidocaine, a common local anesthetic. It's a weak base with a pKa around 7.9. Your blood pH is about 7.4. Since the blood is more acidic than lidocaine's pK, a good chunk of the lidocaine in your blood will actually be in the ionized form.
In this whole thing, drugs changing form based on pH across different body compartments can lead to this phenomenon called ion trapping.
Precisely. Ion trapping is a direct consequence of this. It happens when a drug crosses a membrane in its non-ionized lipid-soluble form.
Slips right across.
Slips right across into a compartment with a different pH. If that new pH causes the drug to become mostly ionized, it suddenly can't cross back easily.
That's trapped.
It's trapped because the ionized form is stuck. A common example, maybe less critical, is weak bases, like opioids accumulating in the very acidic stomach. But a really important clinical example involves the placenta. Many local anesthetics used in labor are weak bases. The fetus's blood is normally slightly more acidic than the mother's blood.
Okay.
So the non-ionized local anesthetic crosses the placenta from mom to baby. Once in the more acidic fetal blood, more of it becomes ionized.
That gets trapped in the baby.
Exactly. It can't easily get back to the mom's circulation. This sets up a gradient, pulling even more non-ionized drug across from the mother. And if the baby gets distressed, their blood becomes even more acidic, acidosis, which traps even more drug. It can become a dangerous cycle.
That's a really powerful illustration of how basic chemistry impacts physiology. Okay, shifting gears slightly, we often hear about the first pass effect, especially with pills we take. What exactly is happening there?
Right. The first pass hepatic effect. This applies specifically to drugs taken orally. When you swallow a pill, it gets absorbed from your stomach or intestine into blood vessels. But these vessels don't go straight into your general circulation. They collect into the portal vein, which goes directly to
The liver.
the liver. So the drug has to pass through the liver before it gets distributed to the rest of the body. Now, if the liver is really good at metabolizing that particular drug, if it has a high extraction ratio, like we talked about,
It gets chewed up before it even gets out.
a significant fraction can be, yes. Think propranolol, or even lidocaine if you took it orally. A lot gets metabolized on that first pass. This is why the oral dose of such drugs is often much, much higher than the IV dose needed for the same effect. So much is lost before it reaches the systemic circulation.
And this relates to bioavailability.
Directly. Bioavailability is just the fraction of the administered dose that actually reaches the systemic circulation unchanged. For 5e drugs, it's 100% by definition. For oral drugs with a high first pass effect, bioavailability can be quite low.
Okay, one last big concept. When we think about how quickly drugs are cleared, I understand there's sort of two main ways the body systems handle that rate.
Yes, we talk about zero-order versus first-order kinetics. This describes how the rate of elimination relates to the drug concentration. Most drugs, thankfully, follow first-order kinetics. This means the body eliminates a constant fraction or percentage of the drug per unit time. So like 50% per hour or 10% per hour, whatever it is for that drug.
The rate depends on how much drug is there.
Exactly. The more drug there is, the faster it's eliminated, but the fraction removed per hour stays the same. This usually happens because the amount of drug is much less than the capacity of the enzymes metabolizing it. There are plenty of enzymes ready to work.
Okay, that's first order. What's zero order?
Zero-order kinetics is different. This happens when the amount of drug is so high that it completely saturates the metabolic enzymes or elimination pathways. The system is overwhelmed. In this case, the body can only eliminate a constant amount of drug per unit time, regardless of how much is present, like 5 milligrams per hour, period. It can't go any faster because the machinery is already working at full capacity.
Are there common drugs that do this?
Yes, some important ones. Alcohol is a classic example. Also aspirin at higher doses, phenytoin, an anti-seizure med, warfarin, heparin, theophylline. With these drugs, small increases in dose can lead to disproportionately large increases in plasma concentration and potential toxicity because the clearance mechanism is maxed out.
Wow, okay, that was an incredible amount of detail. Just thinking about how a drug gets in, where it goes, how it changes, how it leaves. It really makes you appreciate how complex even giving a simple medication can be.
It really does. And if we try and tie this all together, all these principles we've covered, the kinetics, the dynamics, distribution, V-to-TAT, metabolism, clearance, ionization, the order of kinetics, they are absolutely fundamental. They explain how the specific anesthetics mentioned in our sources, from propofol to sevoflurane, even local anesthetics like lidocaine, actually behave inside your body. Their unique properties, based on these principles, dictate how we dose them, how fast they work, and how long you'll feel their effects. It's the science behind the practice.
So what's the big takeaway for you, the listener, joining us on this deep dive today?
Well, we've really journeyed through this intricate dance of how drugs work, haven't we? From binding to a receptor all the way to final elimination. And understanding these concepts, it's not just about memorizing terms like pKa or VAD. It's about building a solid framework, a way of thinking, to really grasp how these powerful chemicals interact with our biology. And maybe it raises a question for you. Think about, say, an over-the-counter pain reliever you sometimes use. How might its properties? Is it lipophilic? How's it metabolized? How might that influence? How quickly it works for you specifically and how long it lasts? What could make it seem more or less effective on different days?
That's really fascinating stuff to think about. And for those of you who are keen to explore even more, the original sources pointed to some fantastic resources. Podcasts like A.C. Hughesgrack, Anesthesia Guidebook, Beyond the Mask. There are some really handy apps too, like ASA-CoAGs for blood clotting info, PD-Stat for pediatric doses, and great YouTube channels, Dirty Medicine and Ninja Nerd were mentioned. They often have really visual ways of explaining these things. There's just a ton of great material out there if you want to keep digging.
Indeed there is. The key is to keep asking questions, keep making those connections.
Absolutely. Well, until next time, keep that curiosity flowing, and thanks for taking this deep dive with us.